Aujourd'hui d'accueillir Jessica Royer. Je la connais depuis plusieurs années. On a déjà collaboré sur des articles même. Jessica a fait son bac et son DEXI en neuropsychologie à l'Université du Québec en Outaouais. Donc, en 2013, je commençais mon DEXI. Et puis, après son DEXI, puis je ne sais pas si c'était pratiqué un peu, mais en tout cas, tu as décidé de retourner aux études, de faire un Ph.D. Qu'elle va terminer. En fait, elle va le défendre dans les prochaines semaines.
Enfin, déposer
défendre dans les prochains mois. Donc, à McGill, avec Boris Bernard. Donc, elle va nous parler aujourd'hui des travaux qu'elle a fait dans son laboratoire, mais aussi des travaux qu'elle a fait en collaboration avec Bridget Foster, qui est aussi à McGill. Je ne la connais pas.
Oui, elle était à McGill jusqu'à tout récemment, elle a changé pour Duke.
OK. Donc, elle était à McGill, elle est partie. Et elle va nous parler donc de travaux qu'elle a fait en IRM et en électrophysiologie chez des patients neurotypiques et des patients épileptiques, je pense. Puis, elle va nous parler en particulier de son approche là, de multiscale. Jessica est une rédactrice d'articles hors pair. C'est quelque chose que je n'avais jamais vu avant, puis je ne verrai peut-être plus jamais. Là, elle a déjà pour une étudiante au doctorat, c'est sûr que c'est son deuxième. Là. Elle a un CV qui est extrêmement impressionnant, énormément d'articles, vous irez les compter sur Google Scholar. Elle n'a pas eu de mal, évidemment, à se trouver une place pour faire un postdoc. Elle va poursuivre sa carrière académique au postdoc à Leipzig, là où on a, où a vu le jour, en fait, la psychologie expérimentale. C'est le premier laboratoire de psychologie expérimentale, c'est à Leipzig. Donc, elle va aller au Max Planck à Leipzig prochainement. En plus de ses travaux de recherche, en plus d'être une rédactrice d'articles hors pair, elle a contribué à la science ouverte. Donc, vous connaissez tous bien la science ouverte maintenant. Elle a contribué à des jeux de données importants et aussi des outils d'analyse importants. Donc, je suis sûr qu'elle va nous parler un petit peu de ces aspects-là de son travail aujourd'hui. Et avant de laisser parler, vous savez, maintenant, c'est une tradition depuis 3 semaines à peu près, ou un mois. On pose des questions tirées là, du questionnaire de Pivot ou questionnaire de Proust aux conférenciers. Donc, je vais poser quelques questions pour vous apprendre à connaître Jessica. Pour la mettre en confiance, je vais lui poser une question qu'elle-même m'a suggérée avant que je la présente. Quelle est ta région du cerveau préférée?
L'insula. Et vous allez en savoir plus pendant la présentation.
Région impliquée dans le dégoût. Je ne sais pas trop. Bon, maintenant, on passe au questionnaire de Pivot. Votre juron, gros mot ou blasphème favori?
Ça fait vraiment aride.
Ouais.
Vraiment, il faut que je dise ça devant tout le monde.
On a le droit de mentir.
Écoute, travaillant surtout en anglais depuis les quelques dernières années, on m'a entendu lâcher à quelques reprises, des fois un fuck ou un shit, quand les affaires vont pas bien.
On est honnête. C'est bien.
Je m'attendais vraiment pas à cette minute.
Quand vous vous adressez à ChatGPT,
Okay.
c'est pas Pivot. Le vous voyez-vous?
C'est une bonne question, ça. Souvent, mes prompts sont en anglais. Fait que là, déjà, la question du vouvoiement est moins applicable, mais je me suis rendu compte que quand je lui pose des questions, je lui demande « please ». Genre,
OK, c'est un peu le même.
C'est un peu bizarrement poli. Je sais pas pourquoi, mais à un moment donné, je me suis comme attrapée. C'était comme « please reformulate ». J'étais comme « il me semble que c'est un peu…
Est-ce que ça pourrait être dû au principe de précaution, par exemple? Parce qu'on ne sait pas ce que ChatGPT deviendra.
Peut-être, je veux qu'il
On
se
va
souvienne
se
de moi.
souvenir de ceux
Peut-être,
qui sont back.
je veux qu'il se souvienne de moi comme une.
Les dernières questions, je vous promets, puis ensuite, je vais laisser Jessica nous présenter son travail. Donc, on retourne à pivot. Si Dieu existe, qu'aimeriez-vous, après votre mort, entendre vous dire?
Wow. C'est vraiment deep comme question.
Quand même.
Dans le dévoilement de soi. It was all worth it.
OK, merci.
C'est vraiment des bonnes questions, Brise-Glace. Merci d'avoir l'atmosphère. Donc, la présentation va être en anglais. Je vais peut-être juste commencer par dire que je suis vraiment l'honneur de me présenter devant vous. Donc, comme Frédéric a mentionné, j'étais ici il y a maintenant quelques années. Donc, j'avais fait un stage avec Frédéric en 2015. Donc, un petit bout. Mais c'est vraiment le fun d'être de retour. Donc, je vais faire un switch un peu abrupt là, maintenant en anglais. So today I'll be talking to you about the work that I've been doing as part of my Ph.D., where I've mostly focused on interactions between structure and function in the human brain, looking at both healthy populations as well as in different neurological and neurodevelopmental disorders. And I think that looking at this relationship from kind of a multi-cell perspective can really bring some nice insights into mechanisms of brain function and better understanding how complex cognition emerges from the human brain. But before jumping into the actual science, I'd like to kind of give you a little bit of background on what got me here. So throughout my training, I had the opportunity to think about this relationship between brain structure and function from different angles. So I'm a neuropsychologist by training, and my clinical practice mostly focused on psychiatric and neurodevelopmental disorders. But during my clinical training, the research I was conducting actually was very behavioral focused. I mostly used the psychophysics methods to study processes like face recognition. And now I'm doing more computational neuroscience related work during my PhD at McGill with Boris Bernhardt, where I studied principles of structure-function coupling in the human brain. And now I'm interested in applying these methods and these concepts to social neuroscience and also neurodevelopment. And I've been starting this as part of different collaborations with some groups in Germany. And I'll be presenting some preliminary results from this at the end of the talk. So I've been very fortunate to contribute to our current knowledge on structure-function interactions and their relationship to behavior through different empirical and methodological angles. So today I hope to convey how multimodal imaging, I think, is a really unique tool to gain a more cohesive understanding of brain organization. And I'll do this by going over some of the work that we've done targeting regions of the limbic system and also disruptions of the system in temporal lobe epilepsy. And I'd like to present ways in which we can make the work that we're doing on this type of data and its processing more transparent, reproducible, and accessible to different research groups. So we know that the brain is a very complex system, and I like to show this figure to kind of illustrate this concept or this idea. This is from a review paper by Rick Betzel and Annie Bassett, published in Neuroimage, that really shows how brain networks are organized across multiple different spatial temporal scales. And these networks can be analyzed at topological scales that range from individual regions or nodes to the network as a whole. And these individual regions really make up the building blocks of these complex networks that make the brain. And decades of research into neuroscience have really demonstrated how these local properties are intertwined with the larger scale organization of the brain. And I think that this is really key to understanding behavior, as the architecture of the brain will constrain how populations of neurons communicate to regulate how we think, how we feel, how we behave, and kind of make us who we are. So in the brain, going down back to the basics, neural activity will depend on neural architecture. This is really a key assumption in neuroscience. And we can study this architecture using different staining techniques, and this has been done historically for the past decades. And this will help us map the layout of cell bodies and myelinated fibers within the cortical gray matter. And several neuroanatomists since the turn of the 20th century have really shown how different brain regions have different laminar architectures, which suggests that they would support different functions. And more recent work has highlighted how regional variations in laminar architecture is one feature that constrains the topography and the strength of cortical-cortical connections. And one of the most notable examples from this line of work is the structural model, which is defined by Helen Barbas, which really lays out these principles under which two cortical regions should be connected based on the similarity of their laminar architecture. And these postmortem studies really provide exquisite descriptions of the organization of cortical microarchitecture, but they really preclude direct accounts of function in the same individuals because they're based on postmortem specimens. So this is a big limitation if we want to study more direct interactions between brain microarchitecture and function. And MRI has been really instrumental in developing this line of research. So with MRI, we can now study brain organization in terms of its structure, its function, in a large number of individuals at a submillimetric scale and within minutes. So this provides us with data that's sensitive to multiple scales of brain organization, from more focal regional properties to the organization of large-scale networks. So these features make MRI this very versatile tool that will allow us to study healthy and clinical populations and also quantify how information provided by different data modalities might be similar across individuals or across different groups of participants. And a lot of these inter-individual and multimodal approaches rely on cortical segmentation tools that allow us to extract and represent the cortical sheet along this reconstructed surface. So if we zoom in here a little bit, so these surfaces segment the peel and the gray matter boundaries, which is shown here in red, and the gray and the white matter boundary, which is shown in yellow. And both surfaces have the same topology and will allow us to precisely sample some of the properties of brain organization that I'll be presenting in this talk. So in recent years, we've really seen the development and widespread adoption of sequences and contrasts that are sensitive to many different properties of brain organization. And some of these contrasts are sensitive to cortical microstructure and particularly to cortical myelin. So these include quantitative imaging sequences like quantitative T1, as well as more ratio-based contrasts like T1 over T2 that have been popularized as part of large data collection consortia like the Human Connectome Project. And these are some of the contrasts that will be mostly at the center of the work that I'll be presenting today. So these images can be used to approximate intracortical microstructural properties of the entire cortex by sampling the intensity of voxels that intersect these surface reconstructions that I just showed in the previous slide. So these measurements are interesting because they're actually quite stable across individuals. They show consistently relatively higher intensities in primary sensory and motor regions compared to heteromodal association cortex. So they'll allow us to differentiate different regions of the brain. And they allow us to compare the microstructural composition of different regions of the brain and also assess disease-related changes in cortical microstructure. And these images can also provide us with indications on regional laminar elaboration. So by constructing a number of surfaces within the cortical gray matter and sampling the image intensities in this depth-wise manner, we can then recover these local microstructural intensity profiles that can then serve as an individual-specific proxy of intracortical myelin and local laminar architecture. And this is an interesting approach if we would then like to see if regional function relates to its microstructure. So in addition to myelin-sensitive contrast, we can then acquire different measures of brain function in the same individuals. So this can include the computation of different time series-based features across different locations in the cortex, but we can also look at more global network configurations, either from resting state acquisitions or task-related coactivation patterns. So then with these different sequences, it becomes possible to assess the correspondence of function and microstructure in a very individual-specific manner once each of the features have been aligned to the same system. And one interesting way to look at function in the brain is looking at functional connectivity. So functional connectivity just basically maps statistical dependencies or correlations between brain regions and how they're active in a more or less synchronized way. So even if our brain is not actively engaged in a given task, so if I'm just laying in the MRI scanner, not doing anything in particular, we can still capture these spontaneous fluctuations in brain activity. And this is what's illustrated in this figure here with the different colors. So we can see that some regions are kind of activated and deactivated at the same time. And we'll then say that regions are connected if their functional activity tends to go up and down at the same time or in a synchronized way. And we could quantify this coordination by simply correlating the signals over time for different pairs of regions. And if we repeat this for all pairs of regions, then we get this functional connectome matrix, which recapitulates all activity time course correlations between all pairs of regions. But this matrix, even we find it quite interesting, it contains a lot of information. So to relate it effectively to other modalities, we might want to extract some relevant features from it first. And one way to deal with this is by reducing the number of areas that we're comparing by considering all voxels or vertices in a given area as a single unit if they show relatively high homogeneity in the feature that we're analyzing. So in these figures here, the same color patch on the brain would tell us that all the data points in this area are considered one region. And this is a very long-standing approach in neuroscience where we try to define different areas or regions or find these areal boundaries. And more recently, this has been implemented with different quantitative techniques like clustering-based approaches, for instance. So this allows us to see if we can get similar boundaries across different types of features or which boundaries might be more variable across individuals, for example. So in case of functional connectivity, we might want to group areas that are connected to each other and dissociate them to areas to which they're not connected. So this could allow us to further characterize the structural properties of a given network, for instance. And this is exactly what one of my colleagues, Casey Piccola, did in our recently accepted paper in Neuroscience. So Casey focused on the default mode network, which is outlined here in red. So she first depicted the frequency of cortical types within the default mode network by leveraging an established atlas of cortical microstructure defined by von Economou in the 1920s. So of all the functional networks, she found that the default mode contained the most balanced representation of the 3 eulaminate types of cortex, which are commonly associated with processing of sensory information and their progressive integration. And this network also contained more dysgranular and agranular type of cortex, which are often linked more to internally generated processes like memory and affect. And using regression dynamic causal modeling, which is a measure of functional connectivity, but that also allows us to infer the direction of signal flow between regions, Casey could further explore how the default mode network was interacting with other networks. So what she found is that while the DMN or the default mode seems to selectively receive inputs from multiple cortical types, its patterns of outputs are remarkably balanced, and this was found to be significantly more balanced than all other functional communities. These are some of the analyses that are presented in the paper that can kind of show how we can leverage parcellations of brain function and structure to inform our understanding of how these systems operate and interact with one another. And complementing these more boundary-based methods are these macro-scale gradients, which amounts to the application of dimensionality reduction to brain data to derive these more continuous dimensions or components of brain organization. And similar to parcellations, gradients also provide this very compact representation of the data, but these approaches rather emphasize more graded transitions between areas rather than hard boundaries. So if we look at the color axis of this figure at the top, so regions with similar colors on this axis would indicate that they share some property that's contained in the underlying data, but the distance of regions along this color axis is also informative because areas with very distant colors on this axis would indicate strong differentiation between them. So in other words, we kind of generate this new ordering of brain regions based on patterns of underlying feature similarity. So these gradients capture multiple different latent dimensions in the data that we're analyzing, with each dimension explaining different amounts of variance in the input data. So here at the top, I'm showing the axis that explains the most variance in functional connectivity data, but other informative patterns of differentiation between functional connectivity patterns of the cortex can also be recovered, for example, dissociating more visual from somatomotor networks. So if we look a little bit more closely at this first gradient of functional connectivity, so as I said, this is the one that explains the most variance in the functional connectivity data. So this is a very purely data-driven technique, but even if it's very data-driven, the pattern that we observe is not random, so it's actually quite structured. So on the one hand, in blue, we find these regions that are associated with more sensory motor programs that are more unimodal and more strongly linked to perception, action, and the external environment. And on the other hand, in red, we have these regions that are associated more with high-level transmodal functions that are decoupled from the here and now. So with this completely data-driven approach, which here uses functional signals while the participant is not engaged in any particular task, we can summarize these global transitions that reflect the hierarchical organization of cognitive systems across the cortex. And we recently applied this approach to intracranial EEG data to better understand the topography of neural dynamics across the cortex. So for this, we use the MNI Open Intracranial EEG Atlas, which is an open data set that gives us access to putatively normal human intracranial recordings. And it's available for both wake and sleep data. For this paper, we just used the wake data. So for those of you that aren't familiar with the clinical workup for epilepsy patients, so patients with epilepsy that don't respond to medication will undergo quite extensive investigations to see if they would be suitable candidates for surgery to remove the part of the brain that's generating their seizures. So for this, we have to try and localize a focal epileptic generator and ultimately try to remove it from the brain to arrest the patient's seizures. And when non-invasive investigations yield inconclusive or conflicting results, then patients are sometimes referred for more invasive studies, where electrodes would be implanted in the brain in select locations to directly record brain activity where we think that the seizures might be coming from or might be spreading to. And this gives us a very high level of spatial precision. But at the single patient level, our coverage of the brain with this technique is very sparse, obviously because we can't implant every region of the brain. It's a very hypothesis-driven technique. But if we put data together from multiple different patients, then the extent of the coverage becomes a lot more interesting. And even if the implantation at a single patient level is hypothesis-driven, so we try to target regions that we think are implicated in activity, sometimes we also implant regions that don't show any implication in seizures. So we think that this is involving relatively normal cortex, or as normal as we can get to with this type of data. So with this data set, we have data from 106 different patients with intractable epilepsy, so totaling over 1,700 data points taken from non-epileptic channels mapped to a common surface template. So looking at the type of power spectrum density shapes that we get in different cortical locations, so we can see that even across this small subset of channels in different locations, the shape of the power spectrum can vary substantially, with peaks occurring at different frequencies in different regions. And we can preserve this interregional heterogeneity in the shape of power spectrum densities when looking either at the channel level or when averaging the power spectrum densities within regions of a 200 region parcellation of the cortex. So then we wanted to derive these gradients of interregional similarity in the intracranial EEG signals to track these more macro scale patterns of similarity in neural dynamics across the cortex. So to do this, we first cross-correlated the parcellated power spectra across all regions that were sampled, and we then derived these gradients of power spectrum similarity across the cortex from the similarity matrix. So with this approach, we obtained these first two gradients that explain the most variance in the data. So this first gradient mostly differentiated for temporal occipital regions from lateral frontal areas, and particularly the precentral gyrus and neighboring regions in blue. And this second gradient that was anchored more in unimodal sensory regions at one end and paralimbic areas at the other. And when we form this kind of 2D representation of neural dynamics formed by these gradients, this really emphasizes the segregation of these unimodal sensory areas in blue from motor areas in pink and paralimbic structures in green. So the topography of this space is in line with foundational models of the organization of the cortical function and microstructure that describes this cortical hierarchy that segregates these perception and action systems, as well as systems that are more tuned to the extrapersonal space from those related to the internal environment. But we don't have to limit ourselves just to functional signals, so we can derive these gradients with any type of data that's represented in a matrix form. So for example, in this paper here, the authors compared functional connectivity, structural connectivity, and microstructural similarity gradients within the same sample of participants. It also doesn't have to be computed across the entire cortex. So when computed at a global scale, it's an interesting approach to assess interregional relationships. But if we do it within a given region, it can also be informative in regards to subregional heterogeneity. So for instance, in this study here, the authors found that in the visual cortex, they could recover the topographic mapping of visual eccentricity and polar angle from local gradients of functional connectivity. And it's also an approach that's not incompatible with hard parcellations, as hard boundaries between neighboring areas can also be recovered from these gradients. So in recent years, we really tried to make these techniques more accessible to the community with different tools and data sharing initiatives. So these efforts have really followed this growing enthusiasm in the neuroimaging community to build more transparent data sharing and methods development practices. And I think that these initiatives have been really instrumental in augmenting our understanding of how the brain's organized across different scales and modalities. So in terms of data, I recently led the development and the release of our multimodal imaging data set, which includes data from 50 healthy controls that were scanned at the Brain Imaging Center at the Neuro. So this first release includes raw and processed data from 4 different data modalities, but also post-processed feature matrices and gradients derived from each of these modalities. So by providing this already pre-processed data, I think that it really facilitates more out-of-the-box use of the data set for studies of structure-function coupling in the healthy brain without the need for a strong expertise in data pre-processing and integrating multiple different data modalities. And recently, our group also released a new precision neuroimaging data set acquired at 7 Tesla. So this is led by my colleague Donna, who's also a PhD student in the lab. So this resource also provides gradients generated from a number of different data modalities, but this time acquired at a higher field strength, and also over multiple sessions in a smaller subset of participants. And alongside my colleague Raul, who's a postdoc in the lab, I also co-led the development and release of our pre-processing pipeline for multimodal MRI. So we originally developed this pipeline to process our own data set, but it's actually very generalizable to any MRI data set that adheres to the brain imaging data structure standards. So the pipeline is called MicaPipe, and it's available on GitHub, and you can run it in a containers with Docker Singularity, and it's also extensively documented. So these resources are part of a broader open neuroscience ecosystem to which myself, but many members of our group have contributed. Some of these target more data sharing, others relate more to processing or analytics, but they all kind of aim to facilitate different aspects of the multimodal imaging pipeline. And I'll just draw your attention to one tool in particular called BrainSpace. So it'll come up a few times in this talk because it's used to generate gradients. So BrainSpace implements different dimensionality reduction methods to recover these continuous spatial trends or gradients of brain organization from any input matrix. So just like MicaPipe, the toolbox is fully equipped with detailed documentation, code examples, tutorials that will help you generate your first gradient if you want to look at gradients. And I'd also like to emphasize how instrumental this toolbox has been and also different data sharing initiatives that have been as well in building a user community around these methods. So this community now gathers gradient enthusiasts from around the world, and we gather at our annual gradients workshop that is organized always right before OHBM. So if you're interested in joining or submitting an abstract, let me know. So now that we have our tools to link cortical structure and function in a meaningful way, I'd like to walk you through some of the recent work that we've done to leverage these techniques and to provide some new insights into principles of structure-function coupling across the cortex. So this paper that I'll be presenting to kind of showcase this is again by my colleague Casey Nicola. I showed you the default mode network work a bit earlier. So Casey is now a group leader in Jülich, Germany at INM7, but this is work that she did as part of her postdoc with Boris. So following what Casey did in this paper, so we keep looking at gradients for this, but kind of shifting our perspective or the data that we're analyzing. So this begins by mapping gradients, not of functional connectivity, but of microstructural similarity across the cortical sheath. So to recover these gradients, we use this really unique data set called Big Brain. So Big Brain is a 3D reconstruction of a Merkur-stained postmortem human brain, where the image intensities directly reflect underlying cytoarchitecture and specifically neural density. So these intensities are sampled across vertices of a number of surfaces that are reconstructed between the peel and the white matter boundaries, and that were segmented specifically for Big Brain. So in doing so, we obtained these microstructural intensity profiles that are shown in this matrix here. And these sample intensities across cortical depths and for all vertices sampled or mapped onto our surface reconstruction. And we can then cross-correlate these profiles across all pairs of regions to quantify how similar our two regions in terms of their intracortical microstructural properties. And we apply dimensionality reduction to recover these spatial gradients of microstructural similarity across the cortex, which this first gradient shown here explains the most variance in the input data. So this resulting gradient will then distinguish regions of the cortex according to the similarity of their intracortical microstructural characteristics. And again, with similar colors in these figures indicating areas that share similar microstructural properties. And regions differentiated along this gradient will follow the overall laminar differentiation of the cortex, as well as these established cytoarchitectonic classes. So this gradient really seems to capture these strong microstructural divergences between more unimodal sensory and motor regions, which show this highly differentiated laminar structure from more limbic areas that show relatively poor laminar elaboration. So using the same approach with myelin-sensitive imaging, so Casey demonstrated this closely replicable topography of this gradient using in vivo MRI, which now unlocks the possibility for individualized depictions of this microstructural gradient in vivo. But then when we correlate these gradients of microstructure and functional connectivity obtained in the same participants, we see this pattern of divergence in certain areas between the two modalities. And when we look a little bit closer at where these regions diverge, so this seems to involve more transmodal cortices implicated in more flexible aspects of cognition. So if we look first at the default mode and frontal parietal network, so these are highly differentiated in their functional connectivity patterns, but they cover a very broad microstructural landscape in the cortex. As a result, they kind of lose their apex position in the topography of the principal microstructural gradient. And limbic regions, on the other hand, are quite unique in their microanatomy relative to sensory motor cortices, but somehow this distinctiveness is not translated into their functional connectivity patterns. And as part of my PhD, I was particularly interested in studying these properties of limbic regions. So I showed you these figures a little bit earlier that demonstrate how limbic areas hold this really unique position at the apex of information processing hierarchies. So information from the external environment is progressively accesses the cortex via more modality-specific sensory cortices, and these low-level features are integrated into more abstract representations harbored in heteromodal and limbic regions. And this hierarchy of information processing across the cortex evolves over these systematic variations in cortical laminar architecture, beginning in highly differentiated sensory cortices and ultimately reaching more dysgranular and agranular limbic areas. And this change in laminar elaboration co-varies with a bunch of other microstructural features of the cortex. And one of these is intracortical myelin content, which is thought to act as a marker of or a force of synaptic stability. So limbic regions are relatively impoverished in intracortical myelin, and this grants them greater plasticity in forming new connections. So this might explain the more kind of flexible patterns of functional connectivity to other regions and the relative decoupling that we see in these areas. And studying these patterns of structure-function relationships within nodes of the limbic system and how this relates to cognitive function has been an important aim of the work that I conducted as part of my PhD. So the insula, circling back to my favorite brain region here. So I think it's my favorite brain region because it's a very nice model system to study properties of structure-function uncoupling in the brain at a smaller scale. So it's kind of a hidden region that interfaces multiple different lobes of the brain, and it shares connections with a bunch of other areas. And this connectivity pattern places the insula as a kind of coordinator of resources between the internal and external environments, and it supports its involvement in sensory, autonomic, and a bunch of social cognitive functions as well. And this functional diversity of the insula is organized in a very structured way. So different studies have highlighted this tripartite division of function within this region. And these divisions generally follow the principal functional connectivity gradient that we find in this area, which evolves along this anterior-posterior axis. And this functional organization is reflected in the insula's heterogeneous microstructure, which also mainly varies along an anterior-posterior axis, but has so far only been studied in postmortem specimens until we came along. So we addressed this gap by kind of mapping these gradients of microstructural similarity across the insular cortex from in vivo proxies of myeloarchitecture using T1/T2 weighted image ratios. So this revealed this anterior-posterior as well as dorsal ventral gradients of microstructural profile similarity in this region's microstructure. And together, these two gradients captured these clear variations in the shape and the mean intensity of laminar profiles across insula subregions. And relating these microstructural gradients to insula functional connectivity, so we find that both gradients anchored these distinct functional transitions across this region. So the posterior insula was mainly connected to multiple sensory systems, but both of the gradients kind of diverge in ventral and dorsal anterior regions to differentiate its affiliations to more attentional and executive systems. So despite this strong microstructural differences between subregions at the extreme of the first microstructural gradient, these two areas kind of seem to share some overlap in their functional connectivity profiles. And in fact, this data kind of suggests that the biggest change that we observe in functional community affiliation in the insula seem to occur along the secondary axis of microstructural differentiation. And if you're interested in more subcortical structures, we also recently developed a way to generalize this microstructural gradient approach to volumetric data. And this is using the amygdala as our proof of principle system. So this is work that was led by a very talented master's student that I co-supervised, Hans Auer, who's now published this work in McGill Life. So Hans used big brain to extract information on the cytoarchitecture of the amygdala. And he built this feature bank of multi-scale intensity variations within the structure. And this allowed him to identify underlying dimensions of variations in these signals that tracked different topographic principles of variability in the neural architecture of the amygdala. So these dimensions could differentiate these known principles of subdivisions of the that we know from from neuroanatomical work done in the amygdala. And this was particularly segregated along the first dimension that we identified in this data-driven analysis of the big brain intensities. And this first dimension was also generally applicable across subjects scanned on our 7 Tesla platform using quantitative T1 imaging. And the amygdala subregions segregated along this first dimension in each participant scanned at 7T could also capture different patterns of functional connectivity, particularly to prefrontal structures. So together, these different studies on the limbic system or limbic subregions show us that we can recover these meaningful variations in functional affiliations of limbic subregions when we anchor our analyses in subregional gradients of microarchitecture. But we also highlight some specific idiosyncrasies of these associations within these systems. So the insula, as I mentioned, we found that these sensory and transmodal changes in connectivity occurred mostly along the secondary axis of microstructural differentiation. And in the amygdala, we could identify a microstructurally and also functionally meaningful first dimension, but the second dimension that I didn't really get into much detail here, it seemed to capture more individual specific patterns of variability that were not reflective of core organizational principles in this region. So moving on to disease applications. So we then asked how microstructural disruptions affecting the limbic system could impact macroscale functional connectivity. And we addressed this by looking at a disease model of altered structure-function coupling that affects a key node of the limbic system with the hippocampus. So temporal lobe epilepsy, or TLE, is the most common drug-resistant focal epilepsy syndrome in adults. And the hallmark lesion associated with this disorder is hippocampal sclerosis, which is defined as loss and paralysis in the hippocampus. And this microstructural feature can be detected using MRI in the form of hippocampal atrophy on T1 weighted scans and signal hyperintensity on T2 flare. And we can use TLE as a disease model of perturbed microstructure function coupling, as this population offers us this really unique framework to investigate how a focal microstructural lesion can affect the organization of large-scale functional systems. So we know that microstructural alterations in the hippocampus particularly affect episodic memory function in these patients. So memory deficits have been kind of repeatedly associated with an atypical organization of memory circuits involving the hippocampus in these patients, but it's also been associated to the degree of hippocampal damage indexed by regional volume reductions, also measured in these patients. And TLE patients also show these more distributed patterns of signal alterations in different myelin-sensitive MRI-based markers. So in this study here, using quantitative T1 imaging, TLE patients showed these significant signal alterations in the mesial temporal lobe, it's ipsilateral to the signal. At more superficial cortical depths, signal changes also extended to more frontal, parietal, and occipital regions. And these findings are also supported by different postmortem studies that show more widespread alterations in the cytoarchitecture of these patients that extend beyond the hippocampus. So overall, these studies support this view of TLE as a more kind of system-level disorder, but these microstructural changes seem to be depth-specific and heterogeneous across regions, and it still remains unclear how impairments in domains relying on more large-scale functional systems like episodic memory might relate to these distributed patterns of microstructural damage in TLE. So we attempted to reconcile these perspectives by first comparing cortical microstructural gradients in TLE to those observed in controls. So the first gradient of cortical microstructure followed similar patterns in both groups by differentiating more primary unimodal and sensory motor regions from paralimbic and paralimbic cortices. But when we compare these patterns more directly with surface-based linear models, we observe overall contraction of the principal microstructural gradient in TLE relative to controls. And we find these significant areas of gradient changes in temporal polar and lateral prefrontal regions, ipsilateral to the seizure focus in patients. So this indicates the presence of changes in the intracortical intensity profiles of these clusters that make them take on certain properties that make them more similar to regions to which they should maximally differ in patients. So overall, these findings suggest these kind of pattern of macro scale de-differentiation of cortical microstructure in TLE relative to controls with significant effects seen in these two areas here. So more temporal polar and lateral frontal. And we implemented a series of different control analyses to assess the robustness of these effects to a multitude of different parameters. And I show one here. So this is looking at when we control for cortical thickness, we also control for regional volume reductions and pericortical blurring, which are all common imaging findings in TLE. And the findings were also consistent across patients and were highly lateralizing. So over 80% of patients in our sample showed at least moderate reductions in gradient scores in the regions that I highlighted. And lower z-scores, either significant cluster, could be seen in the ipsilateral versus contralateral hemisphere in over 90% of patients. And we also looked at disease-related transcriptomics and found that the pattern of gene expression related to hippocampal sclerosis strongly localized with the pattern of microstructural gradient changes that we observed in our sample. And this effect was very specific to hippocampal sclerosis, and correlations were not significant with any other epilepsy symptom. And as part of this study, our participants also completed an episodic memory task inside the MRI scanner, which allowed us to explore how microstructural gradient changes related to episodic memory network reorganizations and also recall performance. So here we assessed episodic memory with a simple paired image association task. So we looked at functional connectivity during the encoding phase of the task, and we targeted regions of significant gradient reductions as seeds, which are indicated by these markers here. And this allowed us to explore how these regions with an atypical embedding within the microstructural gradient space interacted with other regions during memory encoding. And we found that both temporal polar and prefrontal seeds showed these distributed connectivity reductions to limbic and default regions. And we also found that recall performance was significantly correlated with the magnitude of microstructural gradient reductions in temporal regions, but not with gradient changes in prefrontal structures. And these dimensional approaches are also useful in mapping atypical developmental trajectories. So we notably applied this in autism spectrum disorder, or ASD. So previous work has shown an overall contraction of the principal functional connectivity gradient in ASD relative to controls, which reflects this overall reduced segregation of lower order sensory and motor systems from transmodal cortices. And these imaging features reflecting this altered functional differentiation was predictive of symptom severity. And these techniques can also profile individual differences in connectivity patterns and test whether the idiosyncrasy of these patterns contributes to disease expression and behavior. So with this approach, we can basically map kind of deformations of this space in terms of individual specific positioning of nodes and how this distance to a normative connectome space would be predictive to symptom severity. But okay, so these macro scale properties seem to be sensitive to symptom dimensions, but we can also ask if they can inform treatment or relate to treatment effects. And as part of my visit to the University of Lübeck at the beginning of the year, I got to work with this really unique data set, which is a multi-center clinical trial where oxytocin was administered to neurotypical controls and ASD adults in this double-blind, placebo-controlled crossover type of protocol. And we found preliminary evidence of interaction effects in reorganizations of this gradient in ASD relative to controls in response to the administration of oxytocin. So areas of significant changes seem to target the posterior insula. And the position of this region along the principal gradient was kind of already shifted in both groups in the placebo condition. And this difference was actually kind of exacerbated following the administration of oxytocin. And there's prior literature linking kind of autism, oxytocin, and impaired interoceptive processes. So for next steps, we'll probably be kind of more exploring the behavioral relevance of these effects in terms of symptom severity. So together, these different projects contribute to our understanding of how brain anatomy, and particularly cortical microstructure, can constrain these intrinsic neural activity patterns and contribute to particular cognitive functions, and also the organization of more large-scale functional systems. So I've contributed to the development of different open data and software tools to kind of streamline these investigations of microstructure function coupling in the human brain. I showed you ways in which we can leverage these resources to better understand principles of structure-function coupling in the healthy brain, and how unique rules for this coupling seem to apply in some systems, notably in the limbic network. And also presented how we can expand this framework in a typical kind of limbic system disease model, known to perturb microstructure function interactions, and where we find that changes in these intrinsic microstructural coordinate systems of the cortex seems to anchor an atypical functional connectivity during memory encoding. And I discussed how we can then expand this framework to study other conditions like atypical neurodevelopment, for instance. So with that, I'd like to thank everyone who contributed to this work, from Mica Lab, and our collaborators for their wonderful insights in interpreting these results. If you're interested in our open science initiatives, I encourage you to check it out. And thank you for your attention.
Merci pour cette présentation très impressionnante. Actuellement, on prend quelques questions, et ensuite on poursuit les questions avec un verre dans
Ouais, je suis comme I'm the only thing between you and wine.
On va voir si on peut poser des questions avant le vin. Questions?
Après avoir vu la section où est-ce qu'il y avait la différenciation entre un cerveau, mettons, neurotypique et un cerveau de quelqu'un qui est sur le spectre de l'autisme, puis de voir qu'il y a vraiment des différences claires, est-ce que ça se peut qu'un jour, il y ait des diagnostics qui soient faits plutôt que de prendre le temps de faire des diagnostics par des neuropsychologues, que ce soit fait directement dans les IRM?
Question super intéressante, avec des implications vraiment importantes. C'est tout un domaine de recherche, tant dans le neurodéveloppement que dans des applications plus psychiatriques aussi, par exemple. Ça serait un peu le rêve de pouvoir faire passer un IRM, soit anatomique ou fonctionnel, pour pouvoir poser ce diagnostic-là, puis se baser sur des critères, on peut dire peut-être plus biologiques que des associations plus comportementales, qui sont non seulement très coûteuses en temps, mais qui capture des domaines de symptômes qui ne sont pas nécessairement spécifiques à une pathologie en particulier. Donc, par exemple, les troubles attentionnels sont généralisés à travers plusieurs conditions. Donc, ce serait un peu le rêve. Je ne sais pas si on en est là encore. En fait, je ne crois pas. Puis, je pense qu'il y a aussi, pour des pathologies neurodéveloppementales ou psychiatriques, il y a toute une dimension aussi qui est un peu phénoménologique, qu'on ne peut pas négliger, puis qui n'est pas encore accessible avec l'IRM. Donc, on peut imaginer passer un IRM, par exemple, à quelqu'un qui est dépressif, puis dire, bah, oui, vous avez la dépression, mais si la personne ne rapporte pas cette expérience phénoménologique-là, il y a comme un peu une déconnexion qui se fait à ce niveau-là. Je pense que ce serait ultimement un objectif qu'on voudrait atteindre, d'améliorer les processus diagnostiques. Peut-être qu'on pourrait aussi entrevoir, est-ce que ça pourrait informer différentes avenues thérapeutiques aussi. Donc, le diagnostic, c'est définitivement une avenue qui serait importante. Je pense que pour le moment, on n'est pas là, mais je pense qu'on approche peut-être. Je pense qu'il y a quand même des considérations d'interprétation, je trouve qu'il faut aussi intégrer dans l'interprétation de ces données. Ça répond à la question?
D'autres questions là-bas?
Yeah, just more a clarification question.
Yeah.
For the radar plot that you showed with the different networks.
At the beginning?
Yeah.
This one?
No.
More?
Or,
I have a lot of animations.
Yeah. with functional network. Yeah, this one, yeah, this one. So I wasn't sure. So the blue ones, this is psychoarchitecture. So you combined default mode from the parietal based on their psychoarchitecture.
So here, I'll just go back to the previous slide. So in the same participants, so this is from HCP, Human Connectome Project data. So we generated the microstructural gradient from the T1 over T2 contrast, but then we also had measures of brain function and resting state functional connectivity in the same participants. So we have these two complementary gradients of brain organization that tap into microstructure, but also function. And then here, this is stratified according to different functional communities. Are you familiar with the YO communities, like the functional networks? So there's like a group of 7 functional networks that are quite reproducible across a bunch of different participants. And then we can basically track, here it's the difference in rank of regions between these two gradients, and then see how this rank difference was more positive or more negative across these different functional communities. So the functional communities are actually from a different atlas, but it's just mapped to the same surface template.
So completely not with the linked structures, like some solid network, you have the insular, which makes sense. For example, for pure singular, it's in one network, and then you group them based on the cycle architecture. So it's kind of better.
Yeah, the singular gyrus is a really interesting structure in terms of functional organization because it's quite large, but it also spans like multiple different functional communities. So if we look at the sinus network, there's a part of the anterior cingulate that's included. And the much more anterior part, we do tap into some like DMN type processes, and in the mid cingulate, we're actually going from the parietal. So for the cingulate, it can kind of tap into multiple different functional communities.
Pas d'autres questions, on va poursuivre.
On se voit.
Plus informations. Merci beaucoup encore.